Hi, this is Navyashri Chandraya, and this is the presentation for the Neural Networks Project 2, which is Human Protein Atlas Image Classification. Let's know a little about the competition. Here, the objective is to develop machine learning models capable of classifying mixed patterns of proteins in microscope images. The Human Protein Atlas will use these models to build a tool integrated with a smart microscopy system to identify a protein's location from a high throughput image. Proteins are the doers in the human cell, executing many functions that together enable life. In order to fully understand the complexity of the human cell, models must classify mixed patterns of proteins across a range of different human cells. Images visualizing proteins in cells are commonly used for biomedical research, and these cells could hold the key for the next breakthrough in medicine. However, thanks to the advances in high throughput microscopy, these images are generated at a far greater pace than what can be manually evaluated. Therefore, the need is greater than ever for automating biomedical image analysis to accelerate the understanding of human cells and disease. Again, the goal here is to predict various protein structures in cellular images. There are 28 different target proteins. Multiple proteins can be present in one image. So, what's in the data? The data provided contains 31,072 training samples, each with the following. The first is the labels. There are 28 possible classes, and each sample will have one or more labels. The second is four black and white images of the same sample under four different filters, which are the protein of interest, which is a green filter, the nucleus, which is a blue filter, the microtubules, which is red filter, and endoplasmic reticulum, which is a yellow filter. The green filter therefore shows the shape and location of the protein we wish to classify, and the other filters show cell structures that can serve as landmarks or references. The dataset comprises of 27 different cell types of highly different morphology, which affect the protein patterns of the different organelles. Let's talk about the flow I followed in the project to develop a baseline model briefly. First thing is to import the data from the train.csv and then load the dataset info. Unlike most image labeling tasks where binary or multi-class labeling is considered, in this computation each image can have multiple labels. Multi-class multi-label task has its own specific affecting the design of the model and the loss function. And then create a data generator. Our generator will return the next structure which contains RGB images, grayscale images, and the labels. And then visualize the data. Then split the data into train and validation part with a ratio of 80/20. Create a baseline model using a pre-trained model as it is best suited for multi-label classification. Then compile the model using binary cross entropy as loss. And then train the model and monitor quality based on validation F1 score and validation loss. And then finally, visualizing the plots of loss and accuracy of training and validation data set. Here is the glimpse of the visualized data. The first line is the RGB images. The second line is relevant grayscale images. Now let's have a look at the baseline model. Unlike most image labeling tasks where binary or multi-class labeling is considered, in this computation each image can have multiple labels. Multi-class multi-label task has its own specific affecting the design of the model and the loss function. Usage of a pre-trained model is quite helpful in this case. Here I am using a pre-trained model and transfer learning to try and identify the different types of proteins present in the image. I am using exception version 1 model with weights pre-trained on ImageNet. On ImageNet, this model gets to a top 1 validation accuracy of 0.790 and a top 5 validation accuracy of 0.945. The model I am using will have two exception branches. Each will return 2048 sized vectors and they will multiply before prediction. Here is the setup of the rest of the model which we will actually be training. The input shape is 299 cross 299 cross 3. The weights are pre-trained on ImageNet, and I have used max pooling as it is better in this case compared to averaging, which favors larger shapes. The common output function for multi-label tasks is sigmoid, hence sigmoid is used. I'm using binary cross entropy as loss, and the two metrics considered are accuracy and F1 score. One of the challenges in this computation is the 4-channel input RGBY that limits usage of ImageNet pre-trained models taking RGB input. The most common way to convert RGBY to RGB is just dropping Y channel while keeping RGB without any modification. Coming to the performance metrics I'm considering are accuracy, loss, and F1 score. One of the challenges of this computation is strong data imbalance. Some classes like nucleoplasm are very common, while there are some number of classes like endosomes, lysosomes, and rods and rings which are rare. In addition, in tasks of multi-class and especially in multi-label classification, there is always an issue with data imbalance. If you predict one class out of 10, given the same number of examples per each class, you have one positive versus 9 negative examples. So it is crucial to use a loss function that accounts for it. An F1 metric is used for progress monitoring. Talking about the initial performance, I began with using a light exception model and low resolution images to have a baseline that can be used later to select higher end models and explore the effect of image resolution on the prediction accuracy. It is a little big model with two exception branches, as it is taking a lot of time to train. I trained the initial model with only 4 epochs. The validation F1 score of the baseline model achieved is approximately 0.17 and validation accuracy is approximately 96%. Here are the results for 4 epochs. Here are the plots of loss and accuracy for both training and validation sets. The challenges I see are: it is a little bit big model with two exception branches. One epoch trains around 1 hour, which is a huge time. 4 epochs is not enough and hence need to be trained more. The model currently just uses all 3 channels and includes all labels, although some are exceedingly rare. Better image usage and stratification would definitely help. Results are slightly different from one run to another because F1 macro metric is unstable, and several items of rare classes contribute in the same way as thousands of items of common classes. To conclude, it is only a baseline model on how we can use data. It is not a finished variant. We can improve several things such as 4 epochs is not enough, so we need to train more. Change exception network to another one such as Inception v3, and can add augmentation for the data, and play with merge layer and change it to add or something else that fits well. Right now with this baseline model, I achieve a validation F1 score of approximately 0.17 and a validation accuracy of approximately 96%, but some more rigorous training would definitely improve the performance. Thank you.